Major contribution to culture media. Louis Pasteur is the father of microbiology. He introduced complex culture media. And Robert Koch, the father of bacteriology, he introduced solid culture media. Media and culture. Media, nutrients used to grow organisms outside their natural habitats is called as media. That means the media will contain nutrients for the growth of organism, and this media will help us to cultivate or culture the bacteria in outside their natural habitat. So simply we can say that media is a food for microorganism that we are giving artificially that will help us organism to grow. The culturing is the propagation of microorganism using various media. Or culturing means it's a technique to helps the propagation or growth of the microorganism using various media. And also we can say the culture is called as the growth of the organism. Culture media used to grow bacteria. That means the culture media will contain various type of nutrients. With the help of that nutrient, the bacteria will utilize that nutrient and bacteria will grow well in the culture media. And this culture media can be used to enrich the number of bacteria. Enrich the number of bacteria means we can multiply the number. If we have very small number of organism or bacteria, you're inoculating the organism into the culture media, it will multiply, so the number of the bacteria will increase. And this culture media will provide selective growth. Selective growth means if we need a particular organism, or if you want to cultivate a particular, or if you want to select a particular organism from a group of organism, we have selective culture media is there. We inoculate that group of organism into selective media so that the wanted organism will grow well. So we will get a selective growth of particular or wanted organism. And this culture media, we can differentiate bacteria. We have differential media is there. So if you are inoculating the organism or inoculating the multiple organism into that culture media, by seeing the growth in the culture media, we can differentiate the bacteria into two or more. That will help in the diagnostic purposes and also for the identification of bacteria. Ideal medium. The ideal medium should provide satisfactory growth from small inoculum. That means the medium should support microbial growth even from a minimal number of initial cells. That means if you have a very small number of organisms, if you're inoculating the organism into culture media, the organism have to grow well in that particular media. Rapid growth. The ideal medium should provide rapid growth of the bacteria. That means rapid growth means if the medium should have all the nutrients in optimum level, optimum manner, so that the organism will grow rapidly by utilizing the nutrients which is present in the medium. Ideal medium should be easy to prepare. Easy to prepare means the formulation and the step of the preparation of the medium should be simple so that it is very easy to prepare the culture media. And the medium should be cheap. That means economically it will have to be reliable. And the ideal medium should provide, demonstrate all characters of the organism. So if the medium is well and good or ideal, with that medium we can observe the different characteristics of organism that will help us to identify the organism very easily. Easily reproducibility of ideal medium means the media should provide constant or similar result whenever we are cultivating the organism in that particular media. Basic requirement of culture media are nutrients, mineral salts, accessory growth factors, and suitable pH. Nutrients are energy source, carbon source, and nitrogen source. And this energy, carbon, and nitrogen source will provide the nutrients for the growth and multiplication of bacteria. The mineral salts are sulfate, phosphate, chloride, carbonate, sodium, magnesium, and calcium. The accessory growth factors, the accessory growth factors, we are adding extra for the growth of particular organism. For example, in the case of Salmonella typhi, we are adding tryptophan into the culture media so that the Salmonella typhi can grow well in that particular media. And in the X and B factor for Haemophilus influenza, if we are not inoculating, if we are including the X and B factor in the culture media, the Haemophilus influenza can grow well. And suitable pH, that is optimal pH we have to maintain in the culture media for optimum growth of the organism. Here we are talking about medically important organism. So medically important organism, we have to maintain optimum pH for 7.2 to 7.4. Then only the organism will grow well in that particular culture media. Common ingredients, water. Water is a source for hydrogen and oxygen. We know that the bacterial cell consists of water. About 80% of the bacterial cell consists of water. So water is a very, very, very essential ingredients for the growth multiplication of bacteria. The peptone will provide nitrogen and carbon source for the growth of the bacteria. The meat extract will provide growth factor and inorganic salt for the growth of the multiplication of bacteria. Yeast extract will provide protein, amino acid, carbohydrate, inorganic salt, and this will provide the source for growth factor for the bacteria. Electrolytes. Electrolytes, sodium chloride and other electrolytes will help the maintaining the osmotic pressure. Fermentation compound will provide the source of energy. They are sugars and alcohol. Buffers we are using, that is carbonate and phosphates are the buffer. It will helps to maintain the pH of the medium. We already told that if the pH is not optimal, the organism cannot grow well in that particular pH. So we have to maintain the pH in culture medium. The next one is the agar. 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 Agar is obtained from seaweed. That means it is naturally we are getting from seaweeds. Chemically, the agar contain long chain polysaccharides and it is a solidifying agent. Solidifying agent means it is very, very important. Agar we are using in culture media to make the solid media. That means if you are inoculating this, not inoculating, if you are adding this agar into liquid media, that liquid media will be converted into solid media. For example, we have nutrient growth. Nutrient growth is a liquid medium. If you are inoculating 2 to 5% of this agar into liquid medium, that liquid medium will be converted into solid medium. That is, agar is a solidifying agent. And agar melt at 98 degrees Celsius and set or solidify at 42 degrees Celsius. And another thing is that agar is not degraded by the bacteria. That means the bacteria will not utilize this agar. That agar doesn't contain any nutritional value for the growth and multiplication of the bacteria. Agar is just a solidifying agent. That means we can While adding this agar into liquid medium, the liquid medium can be converted into solid medium. Okay. Classification of culture media. Based on the constituency, we can classify or divide the culture media into liquid medium, semi-solid medium, and solid medium. And depending upon or based upon the oxygen requirement, we can classify the media into aerobic media and anaerobic media. This aerobic media means this aerobic media will help us to grow the aerobic bacteria. Those require oxygen for their growth and multiplication, that we will call it as an aerobic bacteria. This aerobic medium will help us to cultivate the aerobic bacteria. The anaerobic media are this anaerobic media. With the help of this anaerobic media, we can cultivate the anaerobic bacteria. That means those bacteria doesn't require oxygen for their growth and multiplication. And this aerobic media, again, we are dividing into simple media, complex media, synthetic or defined media, and special media. And the special media again divided into enriched media, enrichment media, differential media, selective media, sugar media, and transport media. The anaerobic media are Robertson cooked meat medium and thioglycolate liquid media.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.